Dr. Zheng obtained his PhD from the University of Texas Southwestern Medical Center in 1997, after he'd attended Fudan University in Shanghai, China. He did his postdoctoral fellowship at the Memorial Sloan Kettering Cancer Center and joined the faculty at the Department of Pharmacology at the University of Washington in 2002. He's currently a Howard Hughes Medical Institute investigator and a AAAS fellow. His current research focuses on the structural and functional analyses of ubiquitin ligases, their roles in plant hormone signaling, and their potential for novel therapeutic discovery through targeted protein degradation and molecular glue modalities. Dr. Zheng, it's such a great pleasure to have you. I know your work is of great interest to so many people. We're really delighted to have you here. And then I'll add one thing, that please share all your questions with the Q&A tab that's at the bottom of the Zoom panel instead of the chat modality. The Q&A will allow you to post questions that can be seen and which can then be upvoted for questions of particular interest, which we'll try to address at the end. Please go ahead.
All right. Oh, thank you, Saga, for the very kind introduction. Before I start, I would also like to thank the organizers for the invitation and importantly, really developing this very informative seminar series on targeted protein degradation. I think we all agree that there has never been a more exciting time to be in the protein degradation field, as protein degraders are emerging as a new modality in drug discovery in general. Having worked on protein ubiquitination and degradation since I was a postdoctoral fellow in Nicola Pavlovich lab at the Sloan Kettering Cancer Center, I feel very fortunate to witness and to participate in the growth of the field in the past 25 years. I'm also thrilled now that we have the opportunity to translate our basic research into the development of novel therapeutics. So I have to say that starting from the beginning, whoops, hold on. There's some glitches. Okay. Start from the beginning. I've always been fascinated by how versatile E3 ligases are and their role in regulating diverse cellular functions. And I considered it a huge privilege as a postdoc fellow to actually determine the first structure of a ring type of E3 ligase, Cybal, in complex with an E2 enzyme and a substrate degron, which in this case is from ZAP70, a non-receptor tyrosine kinase with a key role in T cell activation. Now, soon after solving this structure and being inspired by the many pioneer and groundbreaking works published by folks like Steve Alich, Wade Harbour, Ray DeShays, and Michele Pagano, among others, Brenda Shulman, my dear lab mate, and I actually started work on the SCF complex, which eventually led to the first glimpse, I would say, of this prototypical multi-subunit choline ring E3 ligase. And in this composite model, I hope you can appreciate that the phosphorylated P27 degron is shown to be co-recognized by the FOX protein SCIP2, which is also known as FBXL1, together with a small protein called CKS1. The next slide is a model of yet another SCF complex that we were able to develop together with a number of lab members after solving the structure of more FOX proteins. In this case is the SCF beta TRCP, which uses a WD40 beta propeller domain to recognize the double phosphorylated beta catenin degron from the Wnt signaling pathway, of course. The study of SCF soon, of course, led to the discovery of CRL superfamily of E3 ligases, which have at least 250 family members, really underline the importance of these superfamily of multi-subunit E3 ligases in human biology. When I started my own lab, we were lucky enough to actually contribute to the field by identifying and establishing CRL4 using a combination of structural biology and proteomic approaches. By solving the structure of DDB1, back then, the only known protein that has been documented to interact with COL4A, we were able to establish it as an unusual adapter of the choline protein. And we subsequently determined the structure of DDB1, COL4A, RBX1 complex associated with a viral hijacker protein called SV5V, which has been demonstrated by Kurt Horwitz in Northwestern to destabilize STATs proteins, which are the signal transducers in the interferon pathway. Back then, we hypothesized that the viral protein most likely mimic the function of the missing cellular substrate receptors of the DDB1 COL4A machinery. And we collaborated with Stefan and Jack in Randy Moon's lab within our own department to tap-tac purify human DDB1 and COL4A. And lo and behold, we pull out the whole family of WD40 domain proteins, which were subsequently named DCAFs together with Wade Harbour's group. Now, although back then we actually noticed a protein called cereblon in our hit list, it was too early for us to realize how it is connected to yet another ongoing project we had in the lab. So I think these early works really demonstrate that E3 ligases can not only coordinate with post-translational modifications to control the timing and specificity of targeted protein degradation, they're also subject to viral exploitation, where viral proteins can subvert these E3 ligases to evade the innate immune system. But given the very large number of choline E3 ligases, we always had a feeling that we were only seeing the tip of the iceberg. In the next 15 years, we realized that degron do not have to be modified by post-translational modification. For example, the extreme C-terminal carboxy group, together with amazingly simple consensus sequence, can be actually effectively recognized by some of the CRL E3s. Here, in collaboration with Sherry Yan's group from Academia Sinica, we reveal that how a adapter protein called CalcDC2 of the CRL2 complex recognize a diglycine C-terminal CN degron, which is generated by premature termination of a translation product. So now, do substrate proteins always need a degron in the form of a short linear motif to be recognized by CRLs? Well, the answer is no. For example, in our circadian clock system, this transcriptional translational negative feedback loop is arguably actually driven by two SCF complexes. In this pathway, period and cryptochrome, which form a heterodimer to turn their genes expression off, are ubiquitinated by SCF beta-TRCP and another SCF complex, SCF FBXL3, respectively. And without these E3 ligases, this negative feedback loop driven pacemaker will get stalled, obviously. Well, interestingly, beta-TRCP recognized the double phosphorylated degron from period, just like it does to beta-catenin. FBXL3 recognized cryptochrome in a completely different manner. In collaboration with Michele Pagano's group at NYU, we revealed that the arc-shaped leucine-rich repeat domain of FBXL3 wraps around the globular domain of cryptochrome. At the same time, it inserts its C-terminal tail deep into the FAD binding pocket of this clock protein. So in doing so, the bonding between this E3 ligase and the substrate can be competed and regulated by first, the FAD cofactor of cryptochrome, and second, the period protein, which is the obligate binding partner of the clock protein. And these are supported by structures we determined for cryptochrome in complex with either FAD or period, both revealing overlapping interfaces with the FBXL3 binding surface. So these structure features not only allow the clock to be fine-tuned by a metabolite, but also actually have a robust oscillation because of its relationship with the obligate binding partner period. And remarkably, every time we turn to a new E3 ligase, we will find something tremendously fascinating. More than a year ago, we published a story on how yet another FBOX protein, FBXL5, regulates iron homeostasis in response to iron abundance and oxygen. So in this case, the target protein is called IRP2, iron regulatory protein 2, which is a well-known protein that regulates the stability and translation of messenger RNAs encoding genes involved in iron metabolism. Now, this IRP2 is recognized and ubiquitinated by FXL5 only when iron becomes excessive, but also it only does it under normoxia, but not hypoxia condition. How does it work? Well, by reconstituting and determining the cryo-EM structure of FXL5 IRP2 complex, we unexpectedly found the iron sulfur cluster bound to the F-box protein, and it is in close proximity to the E3 substrate interface. This potential surrogate of iron plays a critical role in IRP2 binding and would only work in the oxidized state when there is enough oxygen, suggesting it actually acts as a redox switch embedded in this E3 ligase. As you can tell, once again, the F-box protein does not recognize a simple degron motif per se in the substrate protein, but instead recognizing the entire surface presented by a folded domain of the substrate protein. So besides the post-transducer modification, viral hijacking, the E3 substrate interface is really susceptible to different direct regulation of many cellular factors based on these studies, including metabolite as FID, obligate binding partners such as period, or organometals such as iron sulfur cluster, and oxygen, if not a single electron. Now, one important thing I would like to mention is that based on all these studies, we noticed that the affinity between the CRL E3 and its natural substrate always fall into the nanomolar range. And this high affinity is most likely required due to the cellular concentration of the substrate and the E3s, as well as perhaps the critical first step of attaching a ubiquitin to the substrate, as demonstrated by Rita Shea's group. And of course, in the past 20 years, the most important lesson we learned actually come from a field that I have never paid attention before we get exposed to, and that is plant biology. As a kingdom of life that offers humans food, air, shelter, fuel, and medicine, plants in this case actually lend us a principle, if you will, by demonstrating how Mother Nature uses a ridiculously simple small molecule to control targeted protein degradation. Now, before I actually go over the story, I would like to let us first review the status quo of the field at the turn of the millennium, around 2000. As you all know, in year 2001, Rita Shea and Craig Cruz have raised the concept of PROTACs. As seen in this schematic drawing, they propose to make a bifunctional molecule by fusing a known ligand of a target protein to the double phosphorylated beta-catenin degron, so that the resulting chimera protein can bring beta-TRCP into close proximity of the target protein, ubiquitinated and degraded. While this was a great idea, its implementation in drug discovery in general, however, was hindered by the lack of any E3 binding small molecules back then. In fact, there wasn't much hope that human ubiquitin ligases in general are druggable or ligandable back then. This sentiment was perhaps best reflected in this obituary paper published by NCI in 2005. And the title says us off, Missing the Target, Ubiquitin Ligase Drug Stall. Basically, very few companies or academic group succeeded in finding any effective E3 inhibitory compound. Now, around the same time, in year 2005, there was a groundbreaking funding happening in the plant biology field. I really don't have time today to tell you the amazing story of Auxin, except pointing out that first, it is a master plant hormone regulating almost every aspect of growth and development in the green plants. Second, its discovery can be traced back to Charles Darwin, who published a book titled The Power of Movement in Plants in 1880, 2 years before he died. I highly recommend you to read this book. Third, despite more than a century of inquiries, it wasn't until year 2005 when the receptor of this arguably most important hormone on this planet was discovered and mapped to, surprisingly, a plant SCF complex. And this is an absolutely seminal work from Mark Xtal's group, then back in Indiana University, now located in UCSD. We were, again, truly privileged to actually collaborate with Mark Xtal's group and determine the structural mechanism by which F-box protein called TIR1 perceives auxin and reveals how the hormone promotes the interaction between the E3 ligase and the substrate protein via a simple degron motif. To our surprise, unlike any other known hormones, auxin does not induce any conformational changes in its F-box protein receptor. Neither does it bridge the E3 ligase and the degron as a bifunctional molecule. Instead, auxin, just like a tryptophan amino acid, sits actually in between the two proteins and complementing an otherwise imperfect protein interface. To explain its mechanism of action, which is distinct from allosteric switch and PROTAC, we coined the term molecular glue. Now, back in, based on keyword search, actually, we believed we were the first one using this term on a small molecule to describe unique mechanism of action of auxin. But it wasn't until last year that we realized Dr. Stuart Schreiber in Harvard have actually used this term once to describe chemical inducers of proximity in general. For the rest of the talk, however, I will still use this term as we originally meant for reasons that will become clear, hopefully, in the latter part of my talk. Now, if the auxin story escaped the attention of certain proteins, especially those outside the protein degradation field, the IMiD story certainly caught the attention of a lot of people in biology. While the auxin story tells us that Mother Nature have drugged the SCF complex, quote, quote, for billions of years, the 2010 paper published by Hiroshi Handa certainly suggests that a CRL4 E3 ligase have already been drugged by humans in the clinic. And to our surprise, again, the CRL4 DDB1 cereblon E3 we originally identified turned out to be the target of thalidomide. Not only that, but thalidomide and its derivative can also actually do exactly what auxin does by promoting the interaction between the E3 ligase and the substrate, as elegantly reviewed by Ben Eber's group and Bill Kalin's group back in 2013. Now, different from auxin, however, these so-called immunomodulatory drugs actually are thought to enable or foster neo-interactions, interactions that otherwise won't occur in the absence of the compound. Now, thanks to beautiful structure studies by Nicole Thomas group and Eric Fisher in Basel, and as well as Phil Chamberlain's team at Celgene, we were soon able to see the mechanism of action of IMiDs. Lo and behold, again, the auxin-like molecular glue mechanism was revealed for these clinically used and investigational drugs. Needless to mention, the molecular glue concept has since exploded as fireworks, from sulfonamide CDK12 inhibitor to the latest BCL6 degrader, all elegantly done in Dana-Farber and other places. We started to see waves of small molecule acting as molecular glue to induce targeted protein degradation. Now here, I would like to just emphasize again the necessity of serving the term molecular glue for the MOA of these compounds. But really equally important is to give molecular glue a clear and standardized definition. So what is a molecular glue? Unfortunately, when we coined the term, we didn't define it rigorously. The lack of a clear definition makes some people believe molecular glue and PROTACs are the same thing. In other words, glues are just a linkerless PROTAC. Now, others would swear to God that molecular glue is different from PROTACs. They're monovalent chemical inducers of proximity. They're much smaller than PROTACs and more drug-like, and they can have moderate or no affinity toward one of the protein. Although there's nothing wrong with these statements, to our mind, they appear to be rather vague, loose, and descriptive. Obviously, to more rigorously define molecular glue, we really need to identify the unifying theme of all known glues, which also actually should differentiate glues from PROTACs. So in the next 20 minutes, I will recount a recent paper we published as an attempt to define molecular glue. The story really started when I had a conversation with Dr. Liang-Cai Gu in our department of biochemistry. So Liang-Cai did his postdoc work with George Church in Harvard. And in his own lab, part of his interest lie in engineering dual nanobody chemical sensors. Okay, this idea is that he wants to develop two nanobodies that would only interact with each other in the presence of a pre-selected small molecule ligand. And the way that he developed these sensors is relatively simple. First, he would immobilize the ligand on beads and select nanobody from a library where the three complementarity determining region or CDR loops are randomly mutated in this on the nanobody template. After he found a binder, which is called a anchor binder, he would then immobilize this binder on the beads and then screen the library again in the presence of the ligand. And any positive hits binder would then negatively select it if they show any intrinsic affinity toward the anchor binder without the ligand. And the resulting winner is finally selected as the dimerization partner. So using this method, he actually identified several pairs of nanobodies that can sense cannabidiol or CBD, but not its close ortholog THC. The analog THC. So by looking at his schematic drawing, I started to wonder whether the ligand actually functions as molecular glue in his dual nanobody system. So here I just redrew his figure and make it easier to see. The first nanobody anchor binder actually he identified for CBD is called CA14. And the second nanobody he identified, the dimerization partner, is called DB21. Obviously, just because the system was drawn like a molecular glue mechanism with the small molecule sitting in between the two nanobodies, a CBD does not necessarily have to act like a glue, right? We cannot rule out the possibility that CBD actually acts as the elastic switch that binds to a distal end of the nanobody, changes conformation to allow the second nanobody to bind. So to truly understand the mechanism of action of CBD in this case, we decided to first determine the crystal structure of the ternary complex, which we succeeded. And hopefully you can see from here, the overall structure of this complex shows that the two nanobodies pack against each other in a face-to-face anti-parallel manner. And the overall bonding mode of the three molecule is remarkably similar to the schematic drawing actually Liang-Cai originally made. Because the CBD is indeed sandwiched and sequestered in the middle. Now, consistent with his first step of nanobody selection for the anchor binder, CA14 harbors a well-shaped pocket recognizing the majority of the ligand. By contrast, if you take a look at the other side, the dimerization binders, DB21, only contact CBD through a very shallow surface cavity, if not just a depression, to accommodate perhaps the methyl group at one end of the molecule. With these features reviewed, we next actually used ITC to measure binding constant of ligand to the individual nanobodies. While CBD showed a robust 2 micromolar affinity, actually this number should be 2, to CA14 in this ITC experiment, we did not see any binding signal for the DB21, the dimerization partner. And these are, of course, in agreement with the structure. From this measurement and the structure, we can certainly conclude that CBD does not act as a PROTAC by functional molecule with two separate chemical moieties individually engaging one of the two nanobodies. Questions, however, still remain as whether it acts as a molecular glue. Now, obviously, one way we can do it is just like what we did with Auxin system. We can determine the structure of CA14 in the absence of the other two molecules and see whether CBD induce conformational changes within the nanobody, making it compatible to bind DB21. Well, unfortunately, isolated CA14 refused to get crystallized, so we had to actually turn to an alternative approach. We reasoned that if CBD acts as an allosteric switch, that is, it induces major conformational changes of CA14 and reshaping it to a form that's compatible for DB21 binding, then removing the side chain of DB21 residues that are far away from the protein-protein interaction interface, but in contact with the CBD ligand, should really not impact the effect of the ligand. So on top, we first measure using two orthogonal assays the binding constant of DB21 towards CA14 in the presence of CBD. And we found that the two proteins actually interact with each other in the with a double-digit nanomolar affinity. We then actually carefully chose two amino acids, this phenylalanine 48 and histine 101, which are in contact with CBD, but are far away from other residues in the two nanobodies. And we replaced their aromatic side chain with alanine, and we redid the binding experiment using the two orthogonal assays. Now, interestingly, the affinity between the two nanobody dropped by 50 to 120-fold, suggesting that even if the CBD does induce conformational change in CA14, the allosteric effect itself is not a major driving force for CBD's action. Instead, the interface between CBD and DB21 is a major determinant of the ligand's effect. Okay, so far we focus on the interface between the ligand and the two individual nanobodies. From the structure, I hope you can also appreciate that CA14 and DB21 themselves also have very extensive protein-protein interface, mostly through the CDR regions. Now, an important expectation from allosteric mechanism is that without the ligand, the CDRs would be flexible and incompatible with DB21 binding. And this is in fact what Liang-Tsai predicted. Now, to test whether this is the case, we actually set out to measure the affinity between the two nanobodies in the absence of the ligand. Keep in mind, in Liang-Tsai's final selection process, he should have eliminated dimerization partner that would show any detectable affinity to the ligand-free CA14. Well, to our surprise, using two orthogonal assays, we were able to actually detect an about 20 micromolar affinity between the two nanobodies when no CBD was added. The same is true for those two alanine mutations we made that carve out a portion of the CBD DB21 interface. On the one hand, this is perhaps very surprising to Liang-Tsai that the two nanobodies, after all, do have an intrinsic basal affinity, contrary to his belief. It simply for all of the concentration range of his negative selection experiments. So on the other hand, this is actually in full agreement with a molecular glue mechanism in which the major job of the ligand is not to induce conformational changes within the CA14, but instead, CBD really extends, literally, is the protein-protein interaction interface happening between the two nanobodies and fill up a gap between the two proteinaceous components, just like Auxin does. So taken together, we concluded that CBD in this dual nanobody system indeed acts as a molecular glue. And at this point, you might ask, Ning, why bother going out of your way to prove that CBD acts as a molecular glue in this rather artificial system? Well, the answer is that this system presents a case where we actually had to rigorously characterize the thermodynamic parameter of all interacting pairs, which in turn made us better appreciate how a molecular glue system works. Here is the summary of all the interactions we quantified in this ternary complex system. First of all, the ligand has a 2 micromolar affinity toward the anchor binder, but showed no affinity, measurable affinity to the other nanobody. Second, the 2 nanobody actually display a basal weak affinity of 20 micromolar, consistent with the PPI interface formed between them, reviewed by the crystal structure. Third, the ligand enhanced the basal interaction to reach about 35 nanomolar, which is about 600-fold increase. Now, importantly, even though CBD has no affinity to the dimerization partner DB21 alone, the interface formed between them in the ternary complex is absolutely critical. If we carve out 2 amino acid side chain at this interface, the affinity of the 2 nanobodies return to the micromolar range. So now, having quantified the thermodynamic characteristics of this CBD system, we start to wonder about the numbers for the Auxin system. Looking back, it is rather embarrassing to admit that we actually published the structural basis of Auxin perception mechanism without any detailed binding measurement. So we actually did dig out the reagents and actually did rigorous measurement of all interactions in the Auxin system. And without showing you the experimental details, here's the summary of our results. Turns out Auxin binds tier 1 with affinity of 13 micromolar. And not surprisingly, it does not show any affinity toward the degron peptide of the tier 1 substrate protein, which by itself would not adopt a specific conformation. Importantly, in the absence of Auxin, the peptide also shows an about 20 micromolar affinity toward E3 ligase, which is consistent with the fact that the degron peptide does make multiple contact with the tier 1 beyond the interface with Auxin. Similar to CBD, Auxin enhanced the affinity between the degron and the E3 by 1,000-fold. And after Auxin, of course, our attention naturally turned to other prototypical molecular glue systems, and that is the cereblon IMiD system. Looking up the literature, we know that IMiDs has a much higher affinity to the E3 ligase, around 200 nanomolar, and this is why it's great for PROTACs. The IMiDs are also not expected to have any detectable affinity toward the Neo-Substrates because the glycine loop shared by the Neo-Substrates do not have any binding pocket. But do cereblons show any basal affinity toward the Neo-Substrates? The literature suggests not. Since DDB1 decaf proteins are no stranger to us, we decided to purify the protein and measure make the measurement by ourselves. To our surprise, actually in the absence of IMiD compound, cereblon binds IKZF1 with a stunning 250 nanomolar affinity in the absence, of course, of the compound, as measured again by 2 orthogonal assays. And although CKS1 alpha binds cereblon with a slightly weaker affinity, it still displayed a 2 micromolar KD. Just like the other two glues, pomalidomide and lenalidomide augment these interactions and to reach the no nanomolar range. So this table summarizes the numbers we obtained. Now, I have to say that the significant basal affinity between cereblon and its two well-studied neo-substrates truly surprised me, and they actually shattered my previous false hope that molecular glue can actually enable completely neo interactions from scratch. However, I do have a feeling that perhaps this comes no surprise to at least folks in the eBird lab. In fact, if we go back to a supplemental figure found in the seminal 2013 Science paper, a well-controlled experiment clearly showed that IKZF3, if not also IKZF1, can be co-IP'd by cereblon in the absence of the compound, IMiD compound. Moreover, of course, in affinity-based proteomic studies published by a research group in Mayo Clinic, IKZF1 was actually identified as a cereblon binding protein in the absence of IMiDs. And in the most recent article, this is actually an article about CK1 alpha. The protein was reported to be stabilized in the CRBN cereblon dependent manner in response to Wnt signal without the involvement of IMiD compound. To substantiate our quantitative understanding of molecular glue, we next extended our table to include more examples. Now, I don't have space to summarize the complete table, but here I would just want to highlight perhaps the only case of a rational prospective molecular glue discovery program published by the Nurix therapeutic team. In this very well executed program, the company aimed at developing a molecular glue compound restoring the affinity of a hypophosphorylated beta-catenin degron toward the F-box protein beta-TRCP. And in this case, the compound actually displayed no affinity not only to the degron peptide, but also the F-box protein. And this is compensated by the 6 nanomolar basal affinity between the degron peptide and the E3. Remarkably, if you look at the number, the molecular glue compound they developed actually enhanced the affinity to a level that's beyond the missing phosphate site does. Okay, now we have a rather complete set of thermodynamic measurement for most molecular glue degraders. We believe that we're in the position to formally define molecular glue. There are two important unifying themes of these glues arise from our measurement. First, these compounds lack affinity toward at least one binding partner, one protein partner. Second, in all of these glue systems, the two proteinaceous components do have a measurable low affinity basal, low micromolar basal affinity, which are actually enhanced by the compound to reach the nanomolar range. In essence, these molecular compounds really convert non-productive weak protein-protein interaction into a productive high affinity strong interaction. I would like to further mention that the two unifying themes are actually the two sides of the same coin. The pre-existing basal interaction between the two proteins is necessary for molecular glue to act on because the lack of affinity between the glue and at least one of the proteins. If the basal affinity is absent, the ternary complex would not be able to form. So now we have a rather clear definition of molecular glue. Is it really useful at all? Or this is a pure intellectual exercise? I would argue that it does provide some guidance for future molecular glue discovery. Well, first of all, because molecular glues do not show affinity to at least one protein, it can promote the degradation of non-ligandable target proteins that are intractable to PROTACs. Second, in contrast to PROTACs, how to prospectively discover molecular glue degraders for a pre-selected target is not at all obvious in the TBD field. We all know that it is rather straightforward to design PROTAC for a given target. However, most known molecular glues discovered so far are made retrospectively. If we have a genetically validated disease target, how actually do we find a molecular glue? this is particularly challenging for targets to be degraded by E3 ligase as a Neo-Substrate. So out of 200, 600 E3 ligases, which E3 ligase should we choose? Now, obviously, we can come up with some obvious filters, for example, tissue distribution, subcellular localization, E3 abundance and capacity to match the specific Neo-Substrates. But what else? Are all E3s satisfying these criteria created equal for the target? I don't know about you, but this question actually bothered me for a long, long time. We believe our current study now provide the critical criterion for selecting E3s. That is, we want to prioritizing the E3 ligase with detectable basal weak affinity toward the target that will selectively, then we can select that can significantly enhance the chance of success for screening effective glue compounds. I would also like to highlight that this basal affinity is actually the type of non-productive weak interactions that might exist between the E3 ligase and the target of interest. We believe this type of interaction is really widespread among proteins. They are conventionally considered as non-specific, but they're still partner selective. And they're characterized by KDs in the micromolar range, which is above the concentration of most endogenous proteins. Therefore, these interactions are biologically inconsequential. But once they're pushed into the nanomolar range by a small molecule, they will become productive. And in the case of E3 target interaction, it will lead to target protein degradation. Okay, with that, I want to conclude my talk and thank a long list of students, postdocs, colleagues, and collaborators who have made everything possible. And I want to thank NIH and HHMI for funding. On this slide, I do want to highlight that Dr. Shiyun Cao in our group, a postdoc fellow, really did most of the work in the CBD system. And we actually closely interacted with, I interacted with Michele Pagano's group and also, excuse me, Bruce Clem's group at Hutch to brainstorm about molecular glue concepts. And finally, I actually also want to acknowledge my interactions with Jeff Hager, Jason Perani in Coho Therapeutics, and as well as Lan Huang in Seed Therapeutics, and many other scientists in the private sector, including Lilly and Chimera, for discussion on how to prospectively discover molecular glue. With that, I want to thank you for your attention, and I welcome any comments and questions. Thank you.